Fernàndez-Busquets is the head of the Nanomalaria Joint Unit at the Institute for Bioengineering of Catalonia and the Barcelona Center for International Health Research since 2010. He got his PhD in biochemistry at the University Autònoma de Barcelona, and he has been a visiting scientist at Ciba Geigy in Basel, Switzerland, and a postdoctoral fellow at the Friedrich Miescher Institute, also in Basel, and the Marine Biologist Laboratory in the US from 1993 to 1999. At the University of Barcelona, he was also a postdoc fellow until 2001, when he obtained a Ramon y Cajal grant to start his research on nanomedicine. So Xavi, thank you very much. The floor is yours.
Thank you, Christina, for the introduction. Looking at the title, some of you might be wondering why is Xavi saying that his group is for 20 years at IBEC when everybody knows that IBEC was born in 2006. Actually, if you look in our webpage at our history, there you can find information regarding those early times of IBEC. And actually, the institute was born, or its embryo was formed in 2001, when the two mother groups from the UB and the UPC started attracting some researchers, in particular people who got the Ramon y Cajal fellowships in the first call. And those institutes were working for about 5 years until finally IBEC was established. So now this is what could be this IBEC embryo. I don't know, this might be the 32 cell stage, I don't know. As you see, this is a very compact group of people, or we still are today. This is the original entry of the scientific park before the Helix building was constructed. The entrance was through this street. This is actually the IBEC embryo in the staircase that is leading to what were our original quarters. This is the door that leads to the confocal microscope, and then all until the end was this original laboratory of IBEC. As you see, we had some fun, as it's always in small institutes. We had our Mexican parties, we had our fondies, we had, of course, our calçotadas, which we still have today. We have our dinners. Here you can see a young Pera. He just started his PhD in those years. I don't know what was the discussion this particular day. And this is actually the room where today the confocal microscopes are. These are the PhD students and postdocs of that time, to whom I think we deserve some credit. Those were at the same time, they were exciting times because we were witnessing the birth of our institute, but it was also a difficult moment because we needed to be productive, because we needed to create a critical mass of researchers, projects and publications, so that 5 years later, IBEC could be acknowledged as an institute. So I think we deserve this recognition to all the PhD students and postdocs of those original times. Okay, Nanomalaria, probably you don't see much of us, just we appear like the Halley comet in the annual meeting. Many of you might wonder, where are we? Since 10 years, we are actually downtown in the middle of Eixample. This is the hospital clinic building. This is the Escola Industrial. And we are in the Centra Sterkoplovich, the research center from the clinic hospital. We are in the first floor of this building. You are very welcome to visit a Metro Hospital Clinic, but if you come, please do it one at a time. This is the map of our floor. Look at the scale bar. And this is our space. We have only about 6 linear meters of bench. The space downtown is very crowded and the square meter is very expensive. But anyway, you are very welcome to visit. After this brief appetizer, let's go into the main subject of this talk, which is malaria. Malaria is a disease that is caused by a protozoan parasite of the genus Plasmodium. There are several species of it that affect humans, and its life cycle is very complex. To make the long story short, if we start when an infected female Anopheles mosquito bites, it releases sporozoites which go to the liver. In the liver, they infect hepatocytes, and in this stage they can remain for between days to even years until they are released into the blood circulation to infect red blood cells. All the pathologies and all the symptoms of malaria are derived from the red blood cell infection when the parasite grows inside it, and then it digresses to infect new erythrocytes. Eventually, some of those red blood cells, they commit to develop into gametocytes, which are the precursors of the sexual cells. Those gametocytes, if they are ingested by another female Anopheles during a blood meal, in the mosquito midgut, they will develop into gametes, fecundation will take place, and the motile ookinete will cross the mosquito midgut to form an oocyst, which during about 2 weeks, sporozoites will mature to migrate to the salivary glands of the mosquito and to start again the cycle. So this is a very complex cycle and divided between 2 hosts, the human and the mosquito. Our aim is to provide nanomedical solutions for malaria and also, if we can, for other infectious diseases affecting low-income countries. There is a misconception that nanotechnology cannot be applied to that type of diseases because maybe we think that nanomedicine is something very sophisticated and expensive. Maybe in a few years, as a result of the excellent work of some of IBEC groups, we might have devices like this, small nanomachines. But today, we all know that nanomedicine can be just a matter of polymers and a few molecular elements. Nevertheless, it's true that there is a wealth distribution inequality when we are talking of funding for malaria, not only in the funding for projects, but also the World Health Organization recommends that any medicine for malaria that is created should be at a cost of €1 per curing unit. So you need to cure a child sick with malaria with only €1, which, as you can imagine, is very difficult. Actually, then, we are not facing just inequality. We probably should not be giving everybody the same, like cancer, Alzheimer, and malaria, because then we would be in the same situation. We should somehow compensate for this lost time, and I would say that some diseases would require much more funding to reach equity, which I think is what would be our aim. Then the objective of our research turns around drugs. This red star is the drug that should eventually cure malaria, but this drug, as you know, is not delivered usually as such, but it's ideally it's encapsulated in a nanoparticle, and this nanoparticle usually has some type of targeting element. This targeting element, in which we are working in their development, can be antibodies, can be also heparin, and we are also working on evolving DNA aptamers as targeting elements. The nanocarrier itself can be a polymer or a liposome. And regarding drugs, we started working with existing antimalarial drugs, but recently we started the path of identifying new drug targets and actually of discovering new antimalarial drugs. And finally, the target organism obviously is the human, and in there can be red blood cells or gametocytes, but also, as you will see towards the end of the presentation, we are planning to target the insect vector, in particular oocanids or the mid-gut endothelial cells. And then as a target pathogen, basically Plasmodium, but we are also starting having some interesting results with Leishmaniasis. Okay, regarding our first prototypes, they consisted of a liposomal nanocarrier and as targeting element antibodies, and the aim of our first experiments was this. This would be 2 healthy red blood cells, and this is a red blood cell infected by Plasmodium. Let me tell you that the red blood cell is a very nasty cell to target because it doesn't have any endocytosis, and then it's very tricky to penetrate it. In addition, doing it specifically, you don't want to penetrate just any red blood cell, in principle, just those which are parasitized. So that would be the ideal situation where we would have a liposome that we can label in green. For instance, we recognize the targeting antibody, which carries a drug. And then ideally, we should see the liposome or the antibody which targets it only bound to infected cells and also the drug inside those cells. This is actually what we saw right at the beginning, which was a big surprise. This is a field with several dozen red blood cells. Only one is infected. We know that because it's the only one which has DNA. As you know, erythrocytes do not have a nucleus. And then this infected red blood cell is the one that gets all the liposome, and it's the one also that gets inside it, which is the important thing, all the content, which in this case was quantum dots, just to see it if it was working, our approximation. This has been evolved in the last years, essentially thanks to the work of Arnau Biosca, who recently left our group, and now we can make a wide variety of liposomes where we can encapsulate 1, 2 or more drugs, and we can target them with different types of elements or antibodies. We can make antibodies in different parts of it for their use. Liposomes can be made of saturated or unsaturated lipids, where we can in this way control the rate of drug release. We can have a fast release, we can have a slow release, etc. Regarding the targeting antibody, we can also choose an antibody which targets Plasmodium proteins, which would be the ideal scenario, we might think. But then, as you can see here, Plasmodium antibodies are essentially inside the infected red blood cell, and just from time to time you find some of them which are exposed and that can be used for targeting purposes. Regarding this difficulty, or after seeing that, we decided a bold strategy, which was taking an antigen like glycophorin A, which is present in all red blood cells. In that way, we would be targeting the infected erythrocytes, but of course we would also be targeting the non-infected red blood cell, which we did not know exactly how it would work in the end. These liposomes were very good in the sense that they targeted all the infected cells, and also, which for malaria is very important, as I mentioned, we have always to take into consideration the economic landscape. We don't want any liposomes being wasted and not being used. And then with our best formulation, we achieved an almost 100% of use of the liposomes, which were so expensive to produce. One of the consequences of targeting all red blood cells and not only stages of the parasite where the parasite is infecting the erythrocyte is that within the along the 48-hour cycle of Plasmodium in the red blood cell, we are not only targeting the last hours, as it would be if we were using Plasmodium antigens, But we can also target the early stages where Plasmodium doesn't leave any indication in the red blood cell periphery that it's inside it. This was a very good thing because, as you can see here, when we loaded liposomes made in this way with chloroquine, which is one of our control drugs, free chloroquine at these concentrations was not able to kill the parasite in vitro cultures, but when encapsulated in these liposomes, we had a very quick parasite death, which was very good. And another consequence of targeting all red blood cells, we did not realize this at the beginning, but it's a very good thing, especially taking into account that red blood cells are, they just essentially carry oxygen and CO2, and they don't have much more metabolism. And then we realized that probably if you put into healthy red blood cells antimalarial drugs, this will not affect the capacity of the erythrocyte to transport oxygen and CO2 as it was the case. In this scenario, because all red blood cells had drug, when Plasmodium, those that can escape the infected erythrocytes, invade new red blood cells, they immediately find the drugs and they are more rapidly killed. This indeed was confirmed in vivo assays, and we ended up with a very good liposomal prototype that was able to cure humanized mice infected with the malaria, the human malaria parasite Plasmodium falciparum. Then you might think, okay, they got it. We then went to the pharma industry to try to sell our great product. And what was the answer?
You didn't say the magic word.
Of course, the magic word was cheap. Our liposomes were really expensive. Lipids are expensive, and targeting elements like antibodies are also expensive. Then we started looking for other targeting elements, and then we did some research and found out that the polysaccharide heparin had a very good targeting to infected red blood cells. You can see here we did some force spectroscopy experiments with the atomic force microscope, and you can see that binding to non-infected red blood cells was residual, but heparin had a very strong binding to parasitized red blood cells. In addition, there were no Plasmodium resistances known to heparin, and actually heparin has a mild antimalarial activity, not very high, but significant. And then what we did was to substitute the antibody by heparin. We would have the liposomes with the drugs inside, heparin would be coating it. As you can see here, indeed, heparin has a good targeting to even to the intracellular parasite. And what we got was a type of nanocarrier where first we had a good targeting because of this, then the drug that we loaded inside was killing the parasite, and in addition we had the antimalarial activity of heparin also contributing to killing the parasite. Then we had a double life for heparin, which was good because it contributed to reduce the cost of the nanocarrier. It was a targeting element, and then it was also an antimalarial agent in itself. We also tried to reduce the cost of the nano formulations by using polymeric nanocarriers, which were much cheaper than antibodies. In this case, this was a work where we worked with nanocarriers that had a sweet ionic nature. To cross the intestinal barrier, the nanoformulations, ideally, they should have a lot of charge, negative and positive charges, but that the neutral, the overall charge is neutral. And this is what we obtain with this type of nanocarriers. Now what I will show you, in this case, there was no antibody needed. We ended up with finding a polymer that by itself had specific targeting to Plasmodium infected erythrocytes. This is a field with non-infected red blood cells. This is the only red blood cell that is infected, as you can see by the DNA stain. And then we added this polymer labeled with rhodamine to the culture. And as you can see, rhodamine accumulates only in the parasitized red blood cell and not in any of the other erythrocytes, which again was a very good thing. Other types of polymers that we were using were of polyamidamine nature. Polyamidamine nanocarriers can be loaded with drugs. They have a decent drug release, slow, along 24 hours. Again, without needing any antibody, we found some polyamidamines that targeted infected red blood cells. They had good in vitro activity, and again, we had very good in vivo activity in mice. As you might guess, when we went again to the pharma industry, very happy because we followed their advice to find something cheaper but that worked the same. The answer was again tapping our shoulder.
You didn't say the magic word.
And the magic word again, safe, was not there. So we were a bit desperate, and we continued working in trying to improve the nanocarriers. What can be cheaper than antibodies and cheaper than heparin? DNA aptamers. As you know, DNA aptamers are single-stranded DNA chains that, because of their 3D folding, they can generate antigen recognition sites like antibodies do. They are much faster and cheaper to produce than antibodies. They can be made in principle against any antigen, and they do not require animals for their generation. We do DNA aptamers through the CELEX procedure, where what you do is, starting with a random oligonucleotide library, you expose it to your antigen. In our case, if this was red blood cells, first we do a cleaning with non-infected erythrocytes. We don't want all those aptamers that recognize non-infected erythrocytes. Those that do not bind these control cells, we expose them to Plasmodium infected erythrocytes. Then we recover only those that bind, and after a number of cycles, generally between 6 and 10, we end up with specific aptamers. As you can see here, we developed a family of aptamers that recognize very specifically Plasmodium infected red blood cells. In some of the cases, we recognize more than 96% of the parasitized erythrocytes and virtually none of the non-parasitized erythrocytes. So this was also a very important step. This is a work that is being done by our postdoctoral fellow, Elena Lantero, I, and our PhD student, Carlota Roca. We can develop aptamers both against cells, but also against specific proteins that we select as therapeutic targets. Then, in our search for new drug targets, we focused our attention about 3 years ago in the machinery that Plasmodium is using to generate extracellular vesicles. These EVs in Plasmodium mediate parasite cell-cell communication, and they induce them into cytogenesis. They are essential for the parasite. And as you might know, the machinery that in higher eukaryotes is involved in the generation of EVs is the endosomal sorting complex required for transport. In higher eukaryotes, this SCART machinery is made of a lot of proteins. The main ones are depicted here, where you have VPS20, which starts binding to the membrane and recruiting VPS32 to form this structure that is strangling the vesicle that is being formed. And finally, VPS24 and VPS2 do the final strangling, and the vesicle is finally released. The problem is that Plasmodium doesn't have VPS20, it doesn't have VPS22, and it doesn't have many of the other accessory proteins that regulate this process in higher eukaryotes. Then what we did, essentially what it happened is that Yunwen Avalos came to the group. Yun is a postdoc in our group, and what she did was to identify the SCART machinery in Plasmodium, and she found that it's formed by a minimal group of 5 or 6 proteins. VPS20 is substituted by a protein called BRO1, and VPS32 is VPS24 is substituted by VPS60. So with this combination of proteins that Jun found, more or less, it can be explained the formation in Plasmodium of multivesicular bodies, of exosomes, and of microvesicles. And just to show you how efficient this system is, what Jun did was to clone BRO1 and VPS32, and in giant unilamellar vesicles, she injected those two proteins in the GUVs. And the idea is, if these proteins were working, what we should see is the formation of vesicles on these GUVs as it happens in the cell. And actually, that's what Jun observed. Now she is injecting BRO1 and VPS32 in the absence of any other element, no ATP, nothing. And then you will see that with time, vesicles will start forming on the membrane of the GUV. Then Jun has characterized very well this system, and what she is doing now is, because we have the suspicion that this machinery is essential for Plasmodium, we are developing DNA aptamers against those proteins, with the hope that they can be therapeutic elements, and we are starting to have some interesting results with that. Our work along the discovery of new drugs for against malaria, actually it started, I would say, in a very funny way. In 2001, when we joined this IBEC embryo, we were actually happily working with neurodegenerative diseases. We were working with AB40 and AB42 fibrillogenesis. We even ended up proposing models for amyloid fibrils. And as I said, we were having a good prospect in this field. But then what happened in 2002 is that the genome sequence of the human malaria parasite Plasmodium falciparum was published. And then when we read that paper, there was something very peculiar in this genome, which is that it had an extremely high AT content, which resulted in a lot of AU-rich RNAs, which ended up in proteins with an abundance of asparagine and glutamine residues, which, as you know, are the smoking gun, a protein that has this high proportion of these amino acids, is a protein with a high propensity to form aggregates. And actually, looking at the Plasmodium proteome, about 25% of Plasmodium proteins are predicted to be prionogenic, compared with only 1% in human. Then we thought that this was not just chance, and that this would be maybe some therapeutic target. And this is the reason why we started working in malaria, with the idea of bringing some nanomedicines of use for this disease. As you know, a protein that is unfolded should fold properly to be active, but this doesn't always happen. And then in some cases, a protein goes out of the folding pathway, and then it ends up as amorphous aggregates or amyloid fibrils that are usually toxic. Then, knowing this high percentage of aggregative proteins in the Plasmodium proteome, we went to see if this also existed in living parasites. And actually, we found aggregated proteins, but in a massive scale, in all the asexual stages, in free merozoites, in male and female gametocytes, in gametes, in zygotes, in oocysts, in oocytes. We still don't know about the liver stage, but we are working on that. And then what we found, and I cannot give you more details right now because of patenting issues, is a drug that targets this protein aggregation. This drug is very specific. Again, as you can see, when we add this drug to mixture of non-infected and infected red blood cells, actually it only binds where Plasmodium is. So it's a theranostic agent actually, because it can also be used for diagnostic purposes. It has a decent IC50 of 90 nanomolar in vitro. It targets, apart from the liver, which we don't know yet, all Plasmodium stages. It has a low in vivo toxicity. It belongs to a chemical family with no antimalarials. This is very important regarding resistance evolution, because if it belongs to a family where other antimalarials are, Plasmodium is very good in adapting its resistant mechanisms to slightly different drugs. Then probably if it would be of one of the families where we have antimalarials today, evolution would evolve, resistance would evolve rapidly. It targets multiple gene products. This is also good regarding evolution of resistance, and it fluoresces when binding Plasmodium, and it's very easy to synthesize, cheap, and in just 2 steps. This is the work of our PhD student, Ines Bolzón-Arnaiz, and we are very hopeful with this work. But then, again, when we were probing the pharma industry with these good results that we were having, we are, I think, reaching a point where we can provide a very good and very cheap nanocarrier. The last contact that I had with this industry was they saying, well, yes, Xavi, you did magnificent work. In these 20 years, you ended up with having very good nanocarriers, but they are for delivery to the human. And then we always have to do clinical assays. And no matter how cheap is your nanocarrier, it will be very expensive after 15 years of clinical research. Then is when we tried our last approach, which is, okay, Plasmodium has its cycle divided between 2 hosts. One is the human, but then half of its cycle happens in the mosquito. So we started a new research path where we try to target Plasmodium in the mosquito vector and not in the human. This was funded by an European project that is about to end, which is called Nanophiles. And the main results of Nanophiles are finding some elements that can be interesting nanocarriers for targeting Plasmodium in the mosquito. If you remember, in the mosquito midgut, we had the fecundation and the fertilization, and then the motile zygote has to move through the mosquito midgut, and then recognition of the endothelial cells is through polymers which are similar to heparin, through some polysaccharides which had some resemblance to heparin. And then we investigated that a little bit, and we found that ookinides, this stage, is actually a good target for heparin. Then what we did is we explored if heparin would be blocking the development of Plasmodium in the mosquito, and for that we took Plasmodium infected mouse, and then we took blood, and in this blood we added or did not add heparin. And then we gave this blood to feed to mosquitoes. This is our system, where this is a beaker. On top of this there is a kitasato with some blood, and then on the bottom it's open, and all these tiny dots that you see are mosquitoes that are feeding on this blood. Control mosquitoes develop oocysts, which is this stage of the parasite that forms after the ookinete has crossed the midgut epithelium. In this case, we were using a Plasmodium strain that expresses GFP when reaching this oocyst stage, and this is the dissection of a mosquito midgut in the control with lots of these oocysts from where sporozoites will develop. When we were adding heparin, look what happens. The production of oocysts was reduced significantly. Then we have identified heparin as a very interesting drug to be delivered to mosquitoes, essentially to cure mosquitoes of malaria. Another interesting aspect that we could target is that when the ookinete crosses the mid-gut endothelial cells of the mosquito, it does it through a transcellular route. It must go into the cell and go through the cell, not between them. Then what we did was to test different types of polymers to see if we could find some of them that would target antimalarial drugs to these endothelial cells. And indeed, one of the polyamidamines that we were using when fed to mosquitoes, and then we developed an antibody against this polyamidamine, and we did the detection. You see, compared to the control, you see that this polyamidamine accumulates in this is the endothelial layer of the mosquito midgut. This polyamidamine accumulates there, so we are hopeful that this can be used also to target antimalarial drugs to the mosquito. How will we deliver drugs to mosquitoes? We don't know for sure yet. Our planning is to use something similar to fruit fly traps. In this case, it would be something cheap, filled with some mosquito attractant, and then the nanocarrier would be there. The idea is not to kill the mosquito, because if you kill an insect like the mosquito, you might also be killing other insects that are essential for, I don't know, pollinators, that are essential for some crops that people are needing. So we don't want to kill mosquitoes. We just want to kill the parasite inside them, and again at a low price. Okay, that much for malaria, and in the last two slides I will present you our more recent incorporation to our research, which is applying the know-how that we obtained with malaria during all this year to other diseases, and then we started with leishmaniasis. This is the work of our PhD student Lucia Roman, and then Lucia is starting to have some interesting results. Leishmania has also two main stages. One is the promastigote, which is extracellular, and which is the one that the sandfly injects into the human or into the mammal host, where the parasite invades macrophages, and there is the amastigote stage. For the moment, Lucia has been looking for new drugs that could target promastigotes, and actually she found a very good one. And guess which one is this drug? Is the same one that is apparently so good for Plasmodium. So apparently we have also some protein aggregation thing going on in Leishmania. We don't know yet why or what is the reason, but probably this drug would be also something that we could use. Then, this is the last slide. Which antimalarial nanocarrier would we push forward? Well, if Bill Gates would be entering now through that door and saying, "Hey, Xavi, how can I help you?" Then optimal funding, probably, obviously targeting humans, maybe with a liposome nanocarrier, although we have some very good polymeric nanocarriers, antibody targeting until we find good aptamers, our new drug, and targeting human red blood cells. Because I don't see anybody entering, I am afraid that real funding will require a polymer nanocarrier, heparin targeting, the new drug, and mosquito ookinase. So finally, let me thank all the people who have been in the group, both the present members and all the about 50 people who during these 20 years have contributed to obtain those results that I have just presented. And thank you very much for your attention.